Enzymes in general, there are thousands of enzymes, so we have to have a good systematic way of organizing those enzymes so that you can have some meaningful classification. And we do that based on enzyme commission system, okay, or just in short, EC. So for every enzyme, it's going to have an EC number, and actually that's not quite true. I should take that back. So EC numbers are assigned for a particular reaction. Let's say primary alcohol go to a primary carbonyl, which is an aldehyde. It's going to be assigned for a reaction. Okay. However, sometimes substrates are going to be specific, and then it will have an EC number. However, some other times you will have a more like a generalized EC number. You'll have an EC number. Let's say, for example, alcohol dehydrogenase is more general. Okay, but there are going to be specific alcohol dehydrogenases that will have a specific EC number. Okay, so EC numbers, I mean, in general, it's just a very good way for us to know what we're talking about. Okay, but don't get too stressed out about, like what EC numbers mean, like what exactly are the 4 digits, okay? I only want you to know the first digit, okay? So the first digit, that is the class of the enzyme, okay? The last 3, they're not very important. Now, the first digit, it's sort of the class of enzymes, and so far we only have 6 classes of enzymes. And they are Oxidoreductase, Transferase, Hydrolase, Lyase, Isomerase, and Ligase. Okay, so we went through sort of very briefly what kind of reactions they do, or a description of the reactions that they do. So today we're going to look at some sort of pictorial view, or I'll give you some examples. So this slide and the next slide, they sort of summarizes, the type of things that we can do with the 6 different classes of enzymes. So let's start with this slide. So you have your 6 classes. You have first oxidoreductase. And oxidoreductase, simply, it does redox reactions. Redox meaning oxidation and reduction. Okay, so it does oxidation reduction. So the substrate for an oxidoreductase will be either reduced or oxidized, of course, depending on the direction. Okay, so simply put, if you draw a little cartoon, what you can say is here the white little ball here, that's reducing equivalent. Okay, meaning essentially electrons. Okay. Which is going to say that's essentially electrons. You have a chemical A that is reduced, okay? And you have a chemical B that's oxidized. And then your oxidoreductase will facilitate the reaction where it passes the electrons to B. Then your product will become A but oxidized, and then B now reduced. So oxidoreductase is exactly just passing electrons. Okay, so they're passing electrons from molecule to molecule. Okay, so they're doing redox reactions. Right, and some examples or some important subclasses are dehydrogenases, oxidases, reductases, monooxygenases, dioxygenases. I mean, these things, we'll get into exactly what they are probably next semester. We'll talk a lot in detail of all of these enzymes. But today, I'm just going to show you simple examples of what kind of enzymes would belong in this class. Okay. Number 2, you have transferase, okay, and transferases, essentially what it does is it just transfers some functional group. It could be a methyl group, it could be an acyl group, it could be amino group, whatever, okay? Just whatever group. And then it's doing a transfer. In a pictorial view, it's simply said you have A and you have C, okay? And then B, this guy's B. B is like a functional group, okay? It's like acetyl group, for example. And transferases, what it will do is they're going to transfer B from A to C, so that at the end, what you get as a product is A by itself and then BC. There are a lot of different transferases that you see here. You have, for example, C1 transferase, glycosyl transferase. Remember, we heard about glycosyl transferase during the sugar-sugar bond formation, right? When you want to form glucose dimer, for example, remember we had that UDP glucose, and then we had a glycosyl transferase, which will transfer the glycosyl or the glucose part from the UDP to another glucose. Okay, so that's in a way transferase. It transfers. An easy way to know if something is a transferase, okay, typically speaking, it's going to have the word transfer inside. Okay, for example, C1 transferase, glycosyl transferase, amino transferase, phosphotransferase. Okay, so then immediately you know it's a transferase. All right, so those two are pretty easy. Now we're going to get to some exciting stuff like hydrolase. Okay, hydrolase does hydrolysis. Hydrolysis meaning water. Okay, now don't get confused with lyase. Here, hydrolase is using water to essentially break something. Okay? So, for example, you have A, you have B, and then you use water to come in, and then you chop up A and B. Okay? And then you get A with the H, B with the OH. Okay? So you use water to chop something off, okay, to break something, right? Best example ever, chymotrypsin, for example, okay? Proteases, right? Proteases are going to be hydrolases because they use water to hydrolyze your protein, okay? So some other important subclasses, you have esterase, so this cuts an ester, okay? So this guy, for example, cut ester into acid and alcohol. You have glycosidase. Glycosidase is something that cuts glucose, right? Glycosidases that cuts sugar bonds. You have peptidase. Peptidase, so it cuts peptides. Amidase, essentially it cuts an amide, right? So in a way, you're going to be like, wait, that's similar to peptidase, right? But sometimes you're going to have a molecule that has an amide at the end. For example, glutamine, right? Glutamine has an amide. So what amidase will do is it's going to hydrolyze, take water, hydrolyze it, and you get acid plus NH3. So what you see is that hydrolases, essentially what they do is to use water to break something. Okay, always use water to break something. Okay, just remember that. Initially you have a molecule, and then when you use water, it splits. Okay, it splits. Right? Those are hydrolases. Right? For example, nucleases, something that breaks DNA. Same thing. Okay? Same thing. So anything that you think, you look at this reaction and you say, this is a hydrolysis. It's an hydrolase. Okay? Lyase. Lyase is always the one that gets students confused. Okay? Because sometimes it's going to involve water. And then you see, water, it must be a hydrolase. No, okay? Sometimes it's actually a lyase, okay? So lyase, essentially, it's to break or lyase to lyse something. Okay? So using a simple diagram, you have here is B and A, and then you just cut it without water, without anything. You just cut it, you just separate it. You separate A from B, and then you get A and B. That's a lyase. Okay? What do you see different? In hydrolase, the fact that A and B can separate is because you added water. You use water to break A and B. All right? But here in lyase, you don't need to do that. You just break. Okay? Now, the tricky part is actually that sometimes A can be water. Okay? So sometimes you can actually just break water out, and that's a lyase. Okay? So don't get too excited when you see, water is in the equation, it must be a hydrolase. No, 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 no. Okay? No. Right? So you have to think about what it is that you're actually reacting. If it's something that's a chemical that you start with, after the reaction, it produces water, and then B, that's a lyase. Next, we have isomerase. Okay, so isomerase is very easy. Right? This is the easy thing. It does isomerization. Okay, so simply, you have A, and then you do isomerization. You have an isomer of A. For example, glucose become galactose. That's an isomerase, right? I mean, it's just making isomerization, okay? Or you go from ketose to aldose. That's an isomerase, okay? So isomerase, the key is what you notice is always one substrate give you one product. That's an easy way to differentiate isomerase from anything else. You start with A, and then you end up with, I don't know, B, whatever. Okay, so you start with one substrate, you end up with one substrate. Typically, that is an isomerase, right? You did not use any other reactant. So no other reactant, just A become B or A prime. Next, we have ligase. Ligase is also something people get a little bit confused. But if you look at the diagram, it's actually fairly simple. Ligase is, in a way, similar to Lyase. In a way. What do I mean? You have A and B, you end up with B and A. Okay? Or actually, in the case of ligase, most common direction for ligase is A plus B give you AB. Now, in most cases, ligase, okay, or ligase catalyzed reaction is unfavorable unless you have ATP. Okay, so ligase is very simple. Things that use ATP to ligate, okay, to glue two things together, to piece two things together. So, for example, DNA polymerase, ligase, right? It uses sort of ATP and then it ligates something. I don't think DNA polymerase is a ligase. That's a bad example. What's a ligase? Ligase is something like you use the energy stored in ATP to catalyze the ligation of two molecules. So ATP becomes ADP, okay, because the energy in ATP is harvested. Okay, so ATP become ADP. And remember that ATP goes to ADP plus PI. Delta G is minus 30 kilojoule per mole, right? So you have a large amount of energy that you use to drive a chemical reaction. So here's some example. Oxidoreductase, you have lactate to pyruvate. You're going to see this reaction like crazy next semester. Lactate is what? When you go exercise, your body produces lactate. How do you produce lactate? You actually produce lactate from this thing called pyruvate. And what do you notice that's different between pyruvate and lactate? Is that pyruvate is a keto group. Lactate is an hydroxyl group. You go from C double bond O to C single bond OH. And then here, you initially have two bonds, and then you got R, 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 R prime. Okay? So you lost one CO bond. What do you get? You get one CH bond. Okay? And CH bonds have a lot of energy inside. Okay? So what happens is that essentially what you did was you took H minus plus H plus. Okay, so by the way, this guy is H2. And then you take this guy and it goes there, this guy goes up, and then this oxygen grabs this guy. Okay, then you have your, let me change the color real quick. You have this H and this H. Okay, so this is a very typical dehydrogenase that you will see. Okay, and dehydrogenase is a type of oxidoreductase. Okay, so in this case, this is lactate dehydrogenase, and it's called dehydrogenase because essentially it's taking lactate, the two hydrogens plus two electrons. So what is two hydrogen with two electrons? Like that. This guy is H2. H2 is hydrogen. Okay? That's why it's called dehydrogenase. Now, something about oxidoreductase that I'm going to tell you. Okay, it's a little trick. Anytime you see NAD become NADH, that's going to be an oxidoreductase because NAD, this guy here, that's an electron carrier. Okay? Or more specifically, we call it a hydride carrier. Okay? It carries an hydride. What is a hydride? Do you guys know? Hydride. H minus is a hydride. Okay? This thing is a hydride. Okay? NADH is the reduced form. NAD is the oxidized form. Okay? So anytime you see in a reaction NAD, that's going to be an oxidoreductase. Okay? Most likely. Unless it's NAD become NADP, that's not an oxidoreductase, right? But anytime you see NAD become NADH or NADH become NAD, that is an oxidoreductase. Something else, okay, is that a lot of times if you see oxygen as a reactant, that is an oxidoreductase, okay? Because think about it, what else are you going to use oxygen for? Okay, oxidation. Right? You will use oxygen to oxidize things. Right? So anytime you see oxygen in the formula, oxidoreductase, or if you see like NAD become NADH, oxidoreductase. Transferase is a transfer of a reaction or a functional group. In this case, we have a transfer of a methyl group or hydroxymethyl group, not exactly a methyl group. Here we have serine. Serine is an amino acid, and if you want to synthesize glycine, which is another amino acid, you actually take serine. Okay, so you turn serine into glycine. And essentially what you have to do is just transfer this hydroxymethyl group to something else. And then what is left is glycine plus a methylated, or not methylated, methanolylated THF. It's a one carbon carrier. For transferase, I will give you another example. I'm going to show you this. What's a good transferase? I'm going to come to my favorite because you're going to see in a second why this is my favorite. Okay. I have this guy is called acetyl-CoA. And just don't mind CoA. CoA is a coenzyme that's very large. So don't worry about it. It's just, this guy is a acetyl carrier. Acetyl is just this, okay? So 2-carbon molecule. So in my research, I make butanol, which is like, you know, it's like fuel, chemical, money, okay? So it's good. So I want to make biobutanol, right? Biobutanol. And the first reaction is this. You take 2 acetyl-CoA and you make, this thing is called acetyl-CoA. And then plus coenzyme A. This thing is called acetyl-CoA or acetyl transferase. Okay? Why? Because what I did essentially is I transferred this acetyl group to this carbon, right? So I transferred it so that I ended up, the new thing is right here. So I added this carbon, right? So this is your transferred group. And then this guy is this guy. So what did I do? I did transfer of an acetyl group. So this guy is acetyl transferase. Okay? So it's a pretty good example. And then in a couple of steps, okay, in the butanol pathway, it's actually, you can find all sorts of different enzymes. Okay, and then next we have hydrolase. Hydrolase, simple. You just want to cut off something, and here you have urease, okay, and you use water. So always the mechanism is actually quite simple. Attack here, pop up, pop back down, this guy leave. In this case, this is actually more complicated than this because you actually release 2 ammonia and 1 CO2. So you use the water to release 2 ammonia. Lyase, here is a good example, pyruvate decarboxylase, which is the enzyme used in making ethanol. Okay? So if you guys like to go out and drink alcohol, legal drinking age in Taiwan is 18, so you guys can drink. So if you go out and drink, and essentially what you do is you have ethanol, right? And have you ever thought about where do you get the ethanol? Ethanol is fermented, okay? It's fermented from sugar. And essentially in the metabolism of fermentation, you will turn pyruvate into acetaldehyde and then subsequently to ethanol. Okay? Now, here pyruvate to acetaldehyde is a lyase. Why? Because you start with A, one substrate, you end up with B and C. Okay? Right? So you lysed pyruvate into acetaldehyde. So you lysed pyruvate into acetaldehyde and CO2. That's a lyase. Isomerase, in this example, here is methylmalonyl-CoA becoming succinyl-CoA. We'll learn about this next semester as well. Here, what I want to do is I want to transfer this carboxylate from this carbon to this carbon. Okay? So then what I can do is I break this bond and then reform a new bond here. And then it becomes succinyl-CoA. What do you notice? You start with one substrate, you end up with one substrate, isomerase. Okay? Give you another example of isomerase. I'm just gonna. Oops. Okay? Whatever, hexose. This is hexose. Okay? So, what if I do this? What changed? This guy, right? The stereochemistry of that guy changed. This is an isomerase. Okay? Right? It changed the stereochemistry. It's an isomerase. All right? Give you something else. What changed? It went from a aldose, this guy is an aldose, to a ketose. Okay? That's what changed. Right? So you can go from, this is hexoaldose, this becomes a hexoketose. Okay? Aldose to ketose, same thing. You know? I start with one substrate, end up with one product. Isomerase. All right? So isomerase, I think it's relatively easy to understand. Right? Last thing, we have a ligase. Oh, okay, so I guess in this case, they use this example as ligase. Here, I want to take pyruvate plus CO2. Now, pyruvate plus CO2. Now, you're going to be like, whoa, that's crazy. Because CO2 is a gas. CO2 is what? Super stable. So, if you want to take something that's super stable, add it onto a molecule, and then this CO2 is a gas. Okay, you want to capture a gas, put it on a molecule. Yeah, is that likely? No. Okay, thermodynamics is not going to be happy because gas wants to stay as in gas. Okay, you don't want to go from gas to something. Right? So in this case, I have to use the energy of ATP hydrolysis to provide me the power to then convert CO2 onto pyruvate, and then you generate this molecule called oxaloacetate. So basically, you can think of it as CO2 with the ATP. Okay, so it's like activated CO2. Okay, and then this activated CO2 can then react with pyruvate, and you form this bond. Okay, and that gives you oxaloacetate. So ligase, it's always you want to use the power of ATP to do something, okay, to react two things together, okay. So two things together. So for example, A plus B, normally it's unfavorable, but your cell still wants to do it. Then it uses ATP hydrolysis energy to drive this reaction, okay. So you can then convert A plus B into C. Nowadays, people like to use websites to do stuff. So we have this website called Brenda. Okay, so you just type in Brenda. If it's not the number one thing that pops up, it's number two. Sometimes number one thing that pops up is not this website because Brenda is a girl's name. Right? So you probably get someone's personal website. I don't know. But Brenda. Brenda is a powerful, powerful database. Okay, it's really, really nice. I am not even kidding. It's really nice. Okay. And it has a lot of information, right? Now, if you just type in Brenda, right, and then on Google, you'll get the site. And you can do many different things. You can search based on substrate. You can search based on enzyme name. You can search based on EC number. And then what you'll get is you'll get a bunch of different hits, and then you can select whichever enzyme that you want to study. I'm going to tell you right now that this is something that is very useful for my research group. Why? Because a lot of times I want to know what kind of enzymes are available to catalyze a particular reaction. So then I can use that reaction to make something. Right? So then I can type in the substrate and then see if enzyme can do that. Brenda will tell me whether or not there's such an enzyme. Okay? Or then a lot of times I can just type in a particular alcohol dehydrogenase and see if I can find it. Okay? Now, there's another really neat tool that Brenda has. It's called this thing, EC Explorer. Okay? Actually, there are a lot of other options on Brenda that I don't even know how to use. Okay? So if you have time, you can go play with it. EC Explorer. If you click on EC Explorer, this is sort of what you get. Okay? So EC Explorer, EC Browser, and then initially it's going to say, you know, class 1, 2, 3, 4, 5, 6, You click on 1, and then you have 1.1, 1.2, da 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 da. You click on 1.1, and then you get to 1.1.1, so on and so forth. You click on this, and then you get 1.1.1.1, and then you get 1.1.1.100 something. Okay, so the list. And then you can go down this list and then see if there are any enzymes that are of interest to you. Okay, and what is really nice is that, for example, if you select 1.1.1.1, okay, so alcohol dehydrogenase, and here it says 143 organisms. So in their database, they have the information for 143 organisms alcohol dehydrogenase. Okay, and then it's also going to show you a couple of different things. For example, other names, what kind of reaction it does, some comments and history of this thing. Okay, and then what's more exciting is after you click into this entry, and you'll get this website with this page that has all the information you can ever dream of. I'm just kidding, I can dream of more. Okay, but you can get a lot of information. For example, the general nomenclature, like the names and stuff, substrate and product, what kind of substrate and what kind of products they can react. Natural substrates, okay? So why is there a difference between substrate and product and then natural substrate? Because that's what we're going to talk about in just a minute with whatever 5 minutes I have left. Sometimes enzymes will catalyze non-specific reactions, okay? So for my job, I use that property a lot, okay? And then you also get things like Km, Kcat, inhibition, activity, pH optimum, pH range, temperature optimum, temperature range, PI. Remember we talked about PI? Okay, you get all sorts of that kind of information, okay? And then they also direct you to a primary research article in which this database got the information from. Okay, so in the future, if you guys are into enzyme business, Brenda is one of your best friends. Very quickly, we have enzymes are specific to their substrates. We know that. Okay, so that's why people propose the lock and key hypothesis. So enzymes are in general very specific. Okay, and that is because substrate size, substrate chirality, substrate functional group, substrate polarity. Okay, all of them have to match at the active site of the enzyme in order for the enzyme to recognize the substrate. So generally speaking, the shape of the substrate is important. Okay, however, make note that while enzymes are specific for its primary substrates, they can have activity towards other non-specific substrates. Even though I say non-specific substrates, they're not going to be very different from its natural substrate. For example, I can have an alcohol dehydrogenase that naturally reacts acetaldehyde to ethanol. This same ADH can probably react propionyl aldehyde to propionyl alcohol or propanol. Okay, so that's what I mean by non-specific. It's specific in that it does the same type of reaction. It's non-specific in that the substrate size may be a little bit different. I say a little bit, okay? It's not going to be huge different. Okay? The same enzyme probably will not react with something that has 20 carbons. Okay? But 2 carbon versus 3 carbon, it's okay. Right? Now, this property is different between different enzymes. So some enzymes are super specific, okay? Meaning they only react with their natural substrate, nothing else. However, some other enzymes are less so. Okay? Some other enzymes can react many different substrates. It depends on the enzyme. How specific is the enzyme? Okay? Some enzymes are specific, some are not. So the ability to react less specifically is oftentimes referred to as the substrate promiscuity. Okay? Meaning it's promiscuous to accepting different substrate. Okay? Why is this concept of substrate promiscuity very important to my research? Now, very briefly about something that I do in my lab is I want to make renewable chemical and fuel. Typically, this is done through fermentation. Right? So next semester we'll talk all about fermentation. Is where you take plant biomass, make it into sugar, and then you use things like E. coli to ferment, okay, to make things. My research, on the other hand, a significant portion of it at least, is I don't want to use E. coli, right? Because I want to directly use CO2 and go through photosynthetic microbe, directly go to chemical. Why is that better? Because I save this step, I save this step, okay? So then your process is in theory more efficient, right? Efficiency would be higher. Now, one of my target, or at least I did this when I was a PhD student, is butanol. Butanol is great, okay? Butanol is a wonderful compound. It has a large market, it's used almost everywhere. You can use it as fuel, you can use it as polymer material, it is a solvent, you can make it into plasticizers, and it has a quite significant market in the world. Currently, butanol is produced from petroleum, and we know that sucks, okay? Petroleum is running out. So we want to use something green, fermentation, or in my case, CO2 to make butanol. So in my research, it's fairly simple. This is cyanobacteria, a bacteria that can use photosynthesis. So it gets the energy it needs from light. In the process, you can fix carbon dioxide. Right? So this is CO2. CO2 comes into the cell, gets fixed into glyceraldehyde 3-phosphate, so whatever. This is central metabolite. My job is then connect central metabolite to butanol. And here we have some reaction pathway that connected to butanol. However, in this project, there was a challenge, a very specific challenge. That is that this enzyme is oxygen sensitive. So O2 kills this enzyme. If oxygen kills this enzyme, cyanobacteria produces oxygen, then you cannot produce butanol. Okay? That's a problem. So what we did is essentially we looked for enzymes that are oxygen stable. Okay? So I want to find an enzyme that's oxygen stable. Right? So I actually went through a lot of different metabolism and metabolic pathways, and in a particular organism called Salmonella. Salmonella 那个沙门氏菌 okay, 就是那个 rotten egg, right? Not rotten, but inside of the egg, you got a lot of Salmonella. Salmonella has this natural process being able to degrade 1,2-PDO. Right? And in the process, it catalyzes this reaction, a 3-carbon aldehyde to a 3-carbon CoA. What I want is this. I want to have 4-carbon and the reverse direction. Okay? So I don't care. I try this enzyme anyways. So I purify the enzyme and I test for the activity based on whether it's 2-carbon, 3-carbon, 4-carbon, 6-carbon, 8-carbon, and so on and so forth. And then I look at the activity of this enzyme. Of course, because the natural function is to convert 3-carbon aldehyde to 3-carbon CoA. So the 3-carbon activity is high, of course. But I'm very, very lucky in that there are some enzymes that have very good C4 activity. This is called substrate promiscuity. It's non-specific, even though this enzyme's natural job is to do C3 reaction. But it just so happens it does C4 reaction. Using this enzyme, I put it into my cyanobacteria, whatever, and then what you can see is I can get a significant improvement in the amount of butanol that I can produce. So then this is essentially what I do. Okay? I make synthetic organism that produce different things. But how do I do that? I have to rely on a lot of different enzymes. These enzymes, even though they're natural, but their natural function may not be what I want. But in my case, I use them anyways. And then this is based on substrate promiscuity. So don't underestimate the benefit that this can give you. Right? Even though enzymes, we want them to be specific, but sometimes for certain applications, we actually want them to be less specific. Okay?